Welcome friends to this new lecture of soil science and technology, and in this lecture we will be trying to finish the cation action capacity and anion capacity where we left in the last lecture, and then we will be trying we will be discussing about a new topic, that is adsorption of pesticides. So, what is the relationship between pH and CEC? Obviously, generally CEC increases with the pH, and the permanent charge of 2:1 type of clay, that is pH dependent charge of humus and allophane, and some 1:1 type of clay hold exchangeable ions. You already know that. So, at the pH is raised, obviously the negative charge on some 1:1 type of silicate clays, allophane, humus, or even iron aluminum oxide increases, and CEC also increases. Because if you remember, the pH dependent charge, when we are increasing the pH, that will basically create the negative charge. So, creation of the negative charge is basically the increasing the CEC, because it is attracting the cations in the soil surface or clay surface. So, as the pH is raised, obviously for 1:1 type of clay, allophane, and even in iron aluminum oxide, there will be increase in CEC. And in alkaline condition, CEC reflects the pH dependent charge as well as the permanent charge, because you know that permanent charge is permanent. However, in case of alkaline condition, just I told you, it will also develop the negative charge, that is pH dependent negative charge. So, ultimately it will be a total of both permanent charge as well as pH dependent charge. So, the determination of CEC is generally done at pH 7 and or 8.2. So, exchangeable cations in field soils, you know the exchangeable ions in the field soil depend upon the climatic condition. Obviously, the iron and aluminum and complex aluminum hydroxy ions and H plus are most prominent in humid region, and calcium, magnesium and sodium dominate soil in low rainfall areas, because in case of highly leached areas, obviously in case of highly leached, warm and humid condition, obviously there will be formation of oxysols. Now, why there will be formation of oxysol? Because there will be iron and aluminum oxides dominance. So, you will see that in humid region, specifically in the warm and humid region, there will be a predominance of iron aluminum ions. However, in the low rainfall areas, obviously in arid region, there will be predominance of different types of salts because of high evaporative demand. And as a result of high evaporative demand, these soils will be much more, I would say, dominated by calcium, magnesium and sodium ions. So, in a given soil, the proportion of the cation action capacity satisfied by a particular cation is termed as a saturation percentage of the cation. So, for example, if 50% of the CEC is satisfied by calcium ions, the exchange complex is said to have a calcium saturation percentage of 50. So, this terminology is especially useful in identifying the relative proportion of sources of acidity and alkalinity in the soil solution. So, cation exchange properties are typical for unamended clay loam surface soil in different climatic regions. So, if you can see one interesting trend, you can see we are giving 4 different conditions. One is warm and humid region, that is represented by altisol, which is highly weathered, one of the highly weathered soils, and then cool and humid region, and then represented by alphisols, then semi-arid region, which is denoted by oostols, and then arid region, that is natargrids. So, if you see a trend, the exchangeable H plus and L3 plus will be continuously reducing from this warm humid region to cool humid to semi arid and arid region. So, 7.5%, 75% to 28% to 0% almost. And exchangeable calcium will show the reverse trend. I mean, you can get 2% in the warm and humid region. However, we are getting 9% in cool and humid region. It is increasing to 17% in semi-arid region and 13% in the arid region. And also exchangeable sodium, which is basically present in alkaline soil, which is another type of which has the pH high pH. So, in this basically this alkaline soil, an exchangeable sodium is present in a trace amount in case of warm and humid regions. It is very less 0.1%. in alphy soil again 4% in ustols, and in arid soil which is dominated by different sodium salts, you will get almost 19%. So, you can see how these dominance of different cations are changing from for a particular type of soil when it is for a for different climatic conditions. So, that shows the importance of climate for soil development, as we have discussed in our earlier lectures of soil. So, cation saturation and nutrient availability, obviously if the percent saturation of some ions are high, then those ions will be easily and rapidly displaceable obviously. Now, influences of complementary ions, obviously the strength of adsorption of common cations or most colloids is generally follow this order. What is the order? That is, aluminum is strongly adsorbed, followed by calcium, then magnesium, then potassium, which is showing the similar adsorption capacity and which is further further more than sodium. So, potassium can be easily replaced by Al3 ions in the acid soil and it will be available for the plants. Okay. So, in the acid soil, aluminum will replace this potassium and this potassium will be available to the plants. So, there are some nutrient antagonisms. So, this is another important aspect. There is some nutrient antagonism that in certain soil cause inhibition of uptake of some cations by plants. For example, sometimes high potassium levels are known to limit the uptake of magnesium. So, that means potassium and magnesium has some nutrient antagonism, even when significant quantities of magnesium are present into the soil. So, that shows the different interaction or influence of different complementary cations in the soil system. So, here you can have a better idea in this diagram. So, basically it is the soil colloid, basically in our case that is clay, and this is a root extending H plus ion to exchange cations on the colloid, and in the cations in the colloid surface you can see there are several types of cations like sodium and then potassium, and they are basically loosely held complementary ions with large oscillation zones. So, if we compare potassium and sodium, sodium has more large oscillation zone. So, this is called the oscillation zone, and in this oscillation zone they are loosely held, this sodium. So, sodium is easily replaceable than that of potassium. So, root and in the another condition, obviously this aluminum and potassium are present. However, these aluminum are more tightly held complementary ions with small oscillation zone than that of potassium. So, in these two conditions you are seeing basically the difference in oscillation zones and ultimately difference in their affinity to the clay colloid. So, the half circles are loosely held with the soil. So, these are the half circles which are basically loosely held with the soil, and the root will take the cations from the soil in exchange with the H plus ions. So, in the left figure the loosely held sodium plus will be easily taken up by the plant, and in the right figure the clay plus will be easily taken up by the plant, and the K plus will be comparatively more vulnerable to be replaced and sent to the soil solution, and K plus will be more available to the plant uptake and leaching. So, this is how the interaction or between complementary cations influence their uptake by different plants. Okay. So, this is now let us consider another important, very important topic, that is percent base saturation or BS. Now, percent base saturation of the CEC occupied by, it is basically the percentage of CEC which is occupied by the basic cations. So, we have calculated already the CEC. Now, if you can calculate what is the percentage of the total CEC which has been occupied, which is contributing, which is being contributed by the basic cations, that is calcium, magnesium and potassium, then we can calculate the percent base saturation. So, basic cations are distinguished from the acid cations H plus and Al3 plus. So, if you can distinguish, basic cations will be calcium, magnesium and potassium, whereas acid cations will be aluminum and H plus. So, at an approximately soil pH of 5.4 or less, Al3 plus is present in a significantly high concentration than that hinders the growth of most of the plant species. and the lower the soil pH, the greater the amount of toxic Al3 plus. So therefore, soil with high percent base saturation are generally more fertile, because when there will be high dominance of Al3 plus, obviously that will create the nutrient toxicity or Al3 or aluminum toxicity. So we don't need that condition. We need a fertile soil, that means that can support the plant growth, that can support the plant growth, that can supply the required nutrient to the plant. So obviously the soil with a high percent base saturation will have more fertility, and they have little or no acid cations Al3 plus that in the toxic to the plant. So these high percent base saturation soil, they have little or no acid cations like Al3 plus that is toxic to the plant growth. So soil with high percent base saturation have a higher pH obviously, therefore they are more buffered against the acid cations from plant roots and soil processes that acidify the soil like nitrification, acid rain, etc. We will discuss nitrification in our coming lectures. And also they can contain greater amounts of essential plant nutrients, cation like potassium, calcium, magnesium for the use by the plants. So these are very important. What is the formula percent base saturation? The percent base saturation is basically calcium plus magnesium plus potassium over total CEC multiplied by 100. So depending on soil pH, the soil base saturation may be a fraction. This is very important. So depending on the soil pH, the soil base saturation or percent base saturation may be a fraction of CEC or approximately equal to the CEC. So in general, if the soil pH is below 7, the base saturation is less than CEC, because there must be some acidic cations. However, at pH 7 or higher, soil, clay mineral and organic matter surface are occupied by basic cations, and thus base saturation will be equal to the CEC in that condition. Okay. So, what are the effect of different colloid types? Difference exists in the tenacity with which several types of colloids will specific cations. So at a given percentage base saturation smectites, at given percentage base saturation smectites which have a high charge density per unit of colloid surface hold calcium more tightly or more strongly than that of kaolinite. So calcium percentage will have to be increased up to a certain percentage to satisfy the need of plant in case of smectites, and kaolinite can supply calcium at relatively lower percentage of base saturation. And obviously the need to add limestone to the two soils will be somewhat differently, partly because of this factor. And we will discuss why we add limestone when we will discuss the soil acidity. Now let us discuss anion exchange. Anion exchange is also same. Anion, it is basically the ability of the soil to exchange anions from its surrounding medium, and anion are held in two major ways. Firstly, they are held by anion adsorption mechanism similar to the which are those responsible for cation exchange. Secondly, they may actually react with surface oxides or hydroxide, forming more definitive inner sphere complexes. Now in the last lectures we have discussed what are the inner sphere complex and outer sphere complex, so I am not going to discuss them in details. So anion adsorption mechanism, the basic principle of anion exchange are similar to those of cation exchange, and the charge on the colloids are positive and the exchange is among negatively charged anions. The positive charge developed due to the pH dependent charge, you know that, and the positive charge is associated with the surface of kaolinite, iron, aluminum oxides and allophane, and attract anions such as sulfate and nitrate. You can see here, the nitrates is first adsorbed to the soil colloid, and which is further getting replaced by another cation, that is chloride. So just in case of cation exchange, equivalent quantities of ammonia, nitrate and chloride are exchanged here. So the reaction can be reversed and nutrient anions so released can be absorbed by the plants. So AEC generally decreases with the increased pH. Obviously, when there will be increased pH, there will be negative charge development. So when there will be negative charge development due to the pH dependent charge, obviously the the AEC or anion exchange capacity will increase. So this is quite with the increase of with the increase of soil pH, there will be development of negative charge. So when there will be development of negative charge, obviously the anion exchange capacity will decrease, and cationation capacity will increase because it will attract more cation and it will repel more anions and attract more cations. So, these graph basically shows that relationship. So, what is the relationship between weathering and CEC and AEC level? Obviously, you can see there are three different conditions, mostly 2:1 type of clays. You can see here three different conditions. In the first condition is a mild weathering condition, there is an intermediate weathering condition, a strong weathering condition. Mild weathering condition obviously 2:1 type of clays, intermediate weathering condition 1:1 type of clays, and strong weathering condition mostly iron aluminum oxide clays. We have already discussed that. So, increasing weathering is basically in this direction. So, and as we are increasing the weathering, obviously the cationation capacity will decrease. Obviously, you will see most cationation capacity in case of 2:1 type of soil, which will further reduce to 1:1 type of soil, and it will reduce to almost 0 in case of iron aluminum oxides, and for annihilation capacity we will see the reverse trend. So, as we are increasing from mild to intermediate to strong weathering condition, the annihilation capacity will further increase. Okay. So, we have completed this cationation capacity topic. Let us start another important topic, that is sorption of pesticides in the soil. So, we will be covering the importance of pesticide sorption, then distribution coefficient, then binding of biomolecules and to clay and humus. So, importance of sorption of pesticides in soil, you know that soil can absorb charged organic ions by either AEC or CEC, because either it is positive or negative, depending on the positive or negative nature of the charged organic ions, it will be absorbed. And sorption can reduce the movement of the groundwater, because when it will be sorbed, obviously it will not leach down to the groundwater, and it can allow time for soil microorganism to break the chemical down to less toxic byproducts. So, when it will be absorbed by the clay colloids, it will be further, the chance of further leaching down to the groundwater will be reduced, and therefore it will get more time for the microorganism to degrade this. And also it can also produce inner sphere complexes. So, it is common for organic compounds to be absorbed within the soil organic colloids by a process called partitioning, and the hydrophobic part will not be absorbed by the moist clays. Now, what is partitioning? Now, this is a very good picture of partitioning process. Now, this method is also known as the extraction method. So, extraction means drawing a compound out of a mixture using a solvent. So, solvent partitioning is, we can call it solvent partitioning is more specific. So, it means compound have a choice of two solvents that they can dissolve in. Some compound dissolve in one solvent and some compound dissolve in the other solvent. That way the compound in the mixture becomes separated in two groups. So, basically you can see here in this first condition, two compounds are dissolved in a solvent, and now we are adding a second solvent which is denoted by these circles, hollow circles. So, we are basically mixing the two different solvents as well as two compounds together in here, and finally when the solvent separates again, the compounds go into one solvent on the other based on their polarity. So, based on their polarity they will separate into two different solvents. So, this is called partitioning process. Okay. So, this is very important for pesticide sorption. How? We will see. So, this partitioning can be quantified using a partitioning coefficient or Kp, which is basically concentration on solid over concentration in the solution. So, obviously you can see there are this graph shows the relationship between CEQ, which is equivalent concentration or and concentration on solid. So, CEQ is basically concentration in solution, and here it is Q is basically concentration on solid. So, basically this line shows the higher Kp and these line shows the lower Kp. So, high Kp means higher sorption obviously, and low Kp means weak sorption. So, high Kp means hydrophobic compounds on organic matter, when hydrophobic compounds like the organic pollutants, like organic pesticides, they are hydrophobic in nature. So, they will not mix well with the water. So, they will mix more with the organic matter into the soil. And water soluble compounds which are hydrophilic that prefers to stay in the solution will be responsible for this low Kp. So, let us move ahead and see what are the organoclase. Now, organoclase is a very important aspect. The hydrated metal cations like calcium that are adsorbed on the surface of the smectites can be replaced with large organic cations and giving rise to what we are termed as organoclase, that is smectite organoclase specifically. Now, such clay surfaces are more friendly towards applied organic compounds, making it possible for the clay to participate in partitioning. Obviously, when these clays are more friendly towards applied organic carbon compounds, it will take part more into the partitioning processes. So, let us see an example of sorption. So, this is a very good example of sorption process experiment. Now, this experiment was done by a gentian violet color solution and in a sandy loam soil with moderate CEC and a sandy soil with negligible CEC. So, these are two different soil, which is sandy loam soil with moderate CEC and this is sandy soil with negligible CEC. So, after we leach these two soil with this gentian violet solution, a clear solution, and we collect the leachate at below in a beaker. So, after leaching we will see a clear solution was found in case of sandy loam soil, and in contrast water drained from the sandy soil was still purple in color in case of sandy soil with the lower CEC. That means higher CEC or moderate CEC, because in the sandy loam soil there was some amount of silt or organic matter. However, in case of sandy soil there is low amount of organic matter, and due to the presence of organic matter in case of sandy loam soil, these organic matter will absorb all the gentian blue solution. So, ultimately it will the clear solution will leach. However, in the second case the total, so there is no change, so further the purple color solution was leached down. So, that shows the importance of sorption by organic matter which is present in the soil. Now, this is a picture of an African field, and you can see after applying, so in this field scientists have applied, the farmers have applied herbicides and the field is still full of weeds. What is the reason behind that? There is a bizarre condition. We are applying the herbicides to kill the weeds, but we still finding that this is full of weeds. So, what happens? The soil has a upper A horizon, what is A horizon, with a low CEC, and a clay subsoil with high CEC, argillic. Now, what is argillic? Argillic is dominated by clay mineral. So, this soil have a upper A horizon with the low CEC and lower argillic horizon with high CEC. So, when they applied all the pesticides were adsorbed by subsoil clay, so that there is no longer available to be taken out by the roots of the weeds. So, as a result of that, the weeds further grow there. So, that also shows the importance of sorption of different organic pesticides by clay and different other component which are present into the soil. Okay. Now, what is distribution coefficient? The tendency of a pesticide or other organic compound to leach into the groundwater is determined by the solubility of the compound and by the ratio of amount of chemical salt by the soil to remain into the solution, and this ratio is basically known as the distribution coefficient or KD. So, KD is basically milligram of chemical salt per kg of soil over milligram of chemical per liter of soil solution. So, the unit of KD, obviously it is liter per kg, and it depends upon the nature of the soil. Obviously, the variation is related mainly to the amount of organic matter or organic carbon into the soil. So, it can also be represented by using a similar ratio. We call it KOC, or other name is organic carbon distribution coefficient. So, KOC is basically mg of chemical salt per kg of organic carbon and mg of chemical per liter of solution. So basically, we are this is the formula of KOC, that is Kd over gram of organic carbon per kg of soil. So let us see, Kd and KOC are used for herbicides and metabolites. One soil which has higher Kd and KOC values will absorb more pesticide obviously, and these high values indicates that chemicals are strongly absorbed by the soil and less susceptible to leaching. So these are some Kd as KOC values for several widely used herbicides like atrazine, diethyl atrazine, metallochlor, and all these things. You can see what are the variation of their Kd and Kc values, and these are very important for their sorption in the soil. Now, binding of biomolecules and to clay and humus is very important. Now, the bond between biomolecules and the colloid is often quite strong, so that the biomolecules cannot be easily removed by washing or exchange reactions. And the initial attraction may be between charged colloidal surface and positively or negatively charged functional groups or biomolecules, and this type of reaction has two environmental impact. Obviously, the bound chemical remains for a longer period in the soil, as the microorganism cannot recognize and react with the target sites. And secondly, some chemicals remain active after adsorption, so toxin remains toxic to the susceptible organism. Enzyme continues to catalyze the reaction. So these are two types of impacts, environmental impacts you can get from this sorption of biomolecules to the clay and humus. Let us see some examples. Bt toxin, you already know that. Now, Bt is basically a toxin produced by a soil microorganism called Bacillus thuringiensis, and basically it is used for organic farming for protecting crops from insect damage. So it is absorbed by soil. So the 2:1 type of clay, Montmorillonite soil, you can see in this graph. Obviously, the 2:1 type of soil like Montmorillonite can absorb this toxin up to 30 to 80% of its mass. So 1:1 type of clay, obviously it is not that efficient because of low charge development, and obviously the adsorption reaction completed within a minute in case of Montmorillonite. Second, let us see DNA. Now, this is a scanning electron micrograph or same image, SEM image of DNA from Bacillus subtilis bound on kaolinite clay, which is represented in the left picture, or Montmorillonite clay, which is shown in the right picture. So the arrow point stands for basically the strands of strands bound to DNA, strands of bound DNA. Now, DNA bound to clay or humus is protected from decomposition, but retains the capability of transferring genetic information to living soil. So this is implication of DNA sorption in the soil. Antibiotics. The antibiotics also adsorbed by soil colloids by the process of CEC. It also has a very high Kd values. After sorption, it develops positively charged sites in the soil, and increasingly research shows that even though sorption to soil colloids may reduce their efficacy somewhat, the soil-borne antibiotics still works against bacteria. And there is evidence that at least some antibiotics can be taken up from soil by food crops and so enter into the human food supply. So it will increase the resistance against the life-saving drugs into the body. So this is the implication. And let us see one example of antibiotic, that is chlorotetracycline. It is the first antibiotic which has been developed now. Adsorption isotherms illustrates the retention of chlorotetracycline or CTC by Montmorillonite and kaolinite as a function of ionic background cation. So you can see the Montmorillonite or smectite. Obviously, Montmorillonite is showing more adsorption, more adsorption than that of kaolinite, and calcium, CTC retention decrease in the presence of calcium nitrate than that of sodium nitrate, obviously because calcium is more competitive with CTC than sodium. So CTC is adsorbed by the soil via CEC. So, you can see in case of presence of sodium nitrate, the adsorption is higher. However, in case of presence of calcium nitrate, the adsorption is somewhat lower, because calcium is more competitive with CTC than sodium. So, these are some complex interaction between the different molecules which is present into the soil, which we apply into the soil, and the components which are already present into the soil. And you can search some literature, you can obviously consult some literature to gain a in-depth knowledge of these Soption characteristics. These are very interesting, and I would suggest you to go and discuss and to consult some literature to gain more in-depth knowledge of this Soption process, which will be giving you more practical perspective of learning soil science and technology. Thank you, guys. We are completing this week 5 of lectures. So, we will be starting the week 6 of lectures from our next lecture. Hope you have understood and learned something new. In this week, we have discussed several important topics of soil chemistry, and we will be also discussing some other important topics of soil chemistry related to the soil silicate clays in the week 6. So, thank you, and let us meet and in our next week's lecture.